started. We'll be looking at one animation. Really, today it's all about what's called the events at the neuromuscular junction. Effectively, how your nervous system tells the muscle to fire. And what's interesting about that is it's a process that involves a lot of steps, and that's what I had written down. All this series of steps, but effectively, again, normally when you look at nerves in lab, my suspicion is you didn't look at nerves more macroscopically and looking at different parts of the brain and stuff like that in cranial nerves, as opposed to on a microscopic basis. A nerve kind of looks very long. And it's really not a great representation, but I'll do it in a very simplistic. Actually, this part, called the dendrite, in the vast majority of nerves is a receiver that receives the signal. The nucleus of the cell, we can't even call it a nucleus, because in the nervous system, nuclei are a giant cluster of what are called cell bodies. So the true nucleus of a microscopic nerve is actually a cell body. It has other names as well. So you looked at the basal nuclei in the brain in lab, I'm sure, and effectively that's just a giant cluster of microscopic individual nerve cells, nuclei all clustered together. And really what happens is almost like there's multiple branches that receive the signal, and there's multiple branches at the terminus of this long transmitter called the axon. And what we're going to be looking at and focusing at, that's what those images that I will eventually be showing you are about, is effectively what happens as one of these axonal branches contacts this sort of, and this is why my bad arm, a very unique structure on a part of a muscle fiber called the motor endpoint. And that, everybody, is known as neuromuscular junction. So you'll see it a little bit better in their images, and I'll start to transmit these images. We have people joining us. Hi. So let's begin. So I'll show you where we kind of left off. Maybe even a slide before that. We'll click it up. So remember, that might have been very much the last slide that I showed you. And this is where we're going to start looking at some of these specifically at different kinds of channels. And that's why I'm trying to do this for two reasons, limitedly. I'm just going to try to cover one major area each day for today, Monday, and Friday, because I don't want you should be focusing most of your studying on the exam. That's really where so I don't want to overwhelm you with material right now. That's there. And so this is called the neuromuscular junction. And so when you talk about a motor nerve, when they say somatic, it's your brain directing voluntarily some part of your body to do something. Soma, and it's another name interesting for the cell body. The soma means body. Okay. And so anything that has to, so most of your nerves that are telling your muscles voluntarily what to do, we classify them as somatic. You're going to see when we look down the road at the very end of the class, we're going to talk about the autonomic nervous system that we don't control. And a lot of times they'll be classified as what are called visceral nerves. So I don't want to confuse the issue. And thread-like describes these very, very well. An axon is very much a long, slender thread, just like you had taken a tiny spool of thread and stretched it out. And it has branches incoming at the nerve itself, at the dendrite that I drew up there, and at the axon, the terminus spreads out like branches on a tree. And so most of these nerves have multiple hundreds, if not thousands of inputs and hundreds and thousands of outputs. That's how extensive the nervous system is. We'll get to that when we do nervous system. For this, we're going to try to be more simplistic in our approach. So effectively, this is a two-nerve circuit. Your brain to the spinal cord, and it could be a cranial nerve, or to the brainstem, but for ease sake, the brain to the spinal cord, and someone going out to one of your muscles, basically telling it to fire. And effectively, it doesn't go to the whole muscle. These individual threads go to basically individual muscle cells, what we call fibers. And you're going to see this, and they're spread all over. It's not like one part of the muscle is the part that fires. We spread it to all different cells to avoid fatigue. So if you constantly were firing the same area of the muscle, it's going to fatigue more quickly. So when we have to when we lift something and we start to lift it and it's heavy and we got to pull more muscles into it and sort of use more strength, basically we're not only firing more muscles, we're firing different areas of muscles to avoid fatigue that goes along with them. And you see this point's going to be emphasized later when you begin to understand the energy requirements and why muscles fatigue. But it's sort of like a little introductory nugget about that. So these long thread-like extensions, we call it axons, and a transmitter is what it is. Receptor and transmitter. It's an electrical signal. And the axon then branches as it enters the muscle, and the branches go to something that they're going to use the terminology motor unit. We're going to avoid that today and look at this. So the axon branches end on a muscle fiber. That is an individual cell. Again, it could be a foot in length, so it can be a pretty long cell. And we call that junction point the neuromuscular junction. And the unique protein, as you're going to see it sitting on, is called the motor end plate. And there'll be multiple branches. So each muscle fiber has one neuromuscular junction with one branch of this, quote, motor neuron. And so this is kind of what it looks like. So here's that motor neuron, and a branch goes to this fiber, and a branch goes to that fiber, and a branch goes to that fiber. And down the road, that's kind of what a motor unit is. To me, it's inherently confusing, and I don't ask questions about it using that term. I want you to know how it works, not, the uniqueness of the terminology, as it were. So we will be looking at all of those. So they describe it. It's hard to read this even for anybody, much less me. Is the region where the motor neuron contacts the skeletal muscle. It consists of multiple terminals and the underlying junctional folds. You're going to see what I tried to draw on the board, and the illustrations I'll be sending tried to show you this. So it ends, and the spot where they contact each other, they contact, but they don't. There's a space. We talked about the joint space. That's what's called a finite space. There's space in there. This is much tinier. Thousands of these would probably fit in a joint space. It's as close as two things could be without coming in contact. It could be nerve to nerve, nerve to muscle, nerve to gland. There's lots of these kinds of clefts. Collectively, we call them a synapse, and we characterize the opening as a synaptic cleft. You may hear someone else giving you a talk about it, even next term, who says synapse. Doesn't matter, same thing. A lot of things, we have a lot of different pronunciations. So imagine two microscopic structures that just barely touch. You could barely see them touching. We're talking about micrometers. We're talking probably about millimeters. And I think micrometers is better. Millions of an inch or millions of a meter. Extraordinarily small space. And because that space is so small, you're going to see that when a neurotransmitter, in this case acetylcholine, as they're showing you here, this particular, and the best thing about it is the only neurotransmitter that goes to the skeletal muscle. That nerve coming from your brain to the spinal cord actually has two neurotransmitters, one telling the muscle to fire and one telling the muscle not to fire. So here, when it comes from the spinal cord to the brainstem and it goes to the muscle, whenever acetylcholine released, it's basically encouraging, it's probably a better word, that particular muscle or that particular fiber to fire. We'll get what that all means in a second. And you're going to see stored within this, the tip, are what are called synaptic vesicles. A synaptic vesicle, a vesicle is any piece of membrane storage. You're studying exocytosis. You should be for this exam. Exocytosis basically is spitting out stuff out of the cell. All that stuff that's getting spit out are little membrane-bound packages. Effectively, they're all vesicles. The lysosome and the peroxisome are vesicles. They just aren't being spit out. They just stay inside the cell. So they're little membrane packages, membrane spheres would probably be the kind of the best way. And they're filled with a chemical granule called acetylcholine. Okay. And you're going to see as we're going to look at the membrane and the proteins in the membrane, and again, the membrane name is a sarcolemma. They're called junctional folds, and they have an amazingly large amount of these receptors. So once this stuff spits out of the nerve, and I'll tell you the way I used to demonstrate that when I used to give teaching demonstration lectures. I'll get into that in a second. Okay, I used a particular way of trying to show it to students and to the people who are listening to the lecture. So these junctional folds contain millions of acetylcholine receptors, and so much of this spits out when the nerve is triggered that there's a very, very large amount of change that occurs. So the junction, the end of the nerve, the terminal, the cleft, the space, and this particular protein that's got this unique folds, it's all about surface area. The analogy that I was referring to, I have to give credit to my daughter, because when you do a teaching demo for like college or university, you're applying for a job. And so you apply for the job, and I was applying for a particular position. This is more than 20 years ago when I really first started to teach this stuff, about almost 25 years ago. And I said, I want to talk about the nervous system and how nerves communicate with other nerves with chemicals and to kind of spit them out. I'm trying to figure out a good demo for this. And my daughter, I was trying to explain to her, understand my daughter has never been inside of college for whatever reason other than to go to a party. I told you that. Just parties. And again, she only was arrested one time. So I feel that she's got a lot, just once. University of Connecticut during their it's like their super week. It's the weekend before finals. You know, it's like the week before finals week, and it's one giant party. So it's like super weekend, whatever they called up at UConn, up in Storrs, Connecticut. Nevertheless, she said, "Dad, it sounds like Twizzlers, like what?" Because I was the Twizzler, the other thing. Oh, pixie sticks. Are you familiar with pixie sticks? See? And what I would do is I get every one of those pixie sticks, and every time the impulse came down the nerve, I'd pull off or tear off the tip or clip it, and I'd boom with my finger, and out came all these different colored powders. It was for different neurotransmitter. Boom, this is acetylcholine. Boom, this is dopamine. You're going to learn the names. You know, boom, this is serotonin. Everybody loved it. So yeah, I did get that particular job, by the way. But and it's interesting, they didn't make me do that here. They liked me right away. So maybe if they saw that demo, they would have thought better of it. Voila. This is what it looks like. Better than what I drew on the board, even though I'll send you the pictures. So there's a ton of mitochondria because there's a lot of energy that's involved in this particular process. These are the synaptic vesicles. They only contain acetylcholine. This is the axon, the terminus, one of those sort of multiple projections, a little knob at the end. And this is there, and it's a pretty reasonable drawing when you look at it microscopically, of this motor end plate. There's lots of these folds, and again, folds are surface area, and these are the receptors. So understand the complexity of this process. The impulse doesn't come down and magically these gets released. The first thing that they do, and I'm going to show you the animation with regard to it, is hit these. These are a different receptor. These are, just like we saw in that excitation, contraction, coupling, these are called voltage-gated channels. They allow calcium to come in from out here into the cell. That calcium triggers the synaptic vesicles fusing and effectively being exocytosed or spitting out all of these granules. It's such a tiny space.
there's a ton of granules and there's a ton of receptors. And that signal gets passed almost instantaneously. So those are all acetylcholine. And so this is a voltage gated channel. And as you will see, they contact on the other side. This is a ligand or chemical gated channel. This is very confusing. So we start, the electrical impulse opens a channel like flipping a switch, and the chemicals are spit out, and they open a channel by contacting it and changing its shape that way. So that's a chemical or ligand, both terms are used interchangeably, gated channel. And rather than having just one thing like calcium go through it, it has two things. It has sodium going in and potassium going out. And since you have more sodium coming in than potassium going out, it's going to change the resting membrane potential, potential around here. You got 3 coming in and 2 going out. Be familiar. You see this 3-to-2 relationship, whatever direction they're going. Down the road, it's going to activate eventually to reset this cell chemically. It's going to activate those sodium potassium pumps. They spit back out the 3 sodiums and pull back in the 2 potassiums to reset it chemically. And I'm using the term advisedly. You'll see why as we move forward. So this is just the junction. And the whole ball game is if we get enough of these triggered to change that resting membrane potential to a number we call threshold, it will cause this signal, basically this whole membrane, to basically flip-flop in the sense of going from positive on the outside to negative on the inside rapidly, chain reaction all across the membrane of the entire muscle fiber. And we call that phenomena an action potential. So it's brought out, confusing enough, by an action potential coming down the nerve. And we hope, if we're going to fire that muscle, by an action potential resulting here at the motor end plate and going through the whole membrane. Confusing enough. Okay, big picture. This is what we're looking at today, the events of neuromuscular junction. I'm going to go over that excitation, contraction, coupling again, and probably introduce the cross-bridge cycle on Monday without going crazy over the top, because I know you'll be busy, hopefully distracted by the other material. This is how complex it is. This is what I wrote. They write it again a little bit better, but without me reading it excessively to you. Here comes the action potential down the nerve. That action potential causes, by, as you're going to see in the animation, causing those gates to open, those voltage gated gates for calcium. It leads to the release of the acetylcholine at the cleft. The acetylcholine receptors are triggered, and this is where terminology is a little confusing. We're trying to achieve an action potential, meaning it will cause that nerve to effectively fire that muscle and have it shorten. But if it does not, we call it an end plate potential. You see that term over here. Our bodies are, all of our muscles are continually being stimulated, whether you knew it or not. We have a thing called muscle tone, resting muscle tone. If someone is comatose, and what defines it almost as if you lift an extremity like it'll flop back down. It's basically, bless you, receiving very, very little in the way of signals. If you have someone who's sleeping and do that, you'll be able to feel that there's tone. In other words, the muscles are not flat or flaccid. They will have, you can feel the tension in them. You can feel that they're kind of loaded in a way to express that. And that's very important. As I was saying to all the other classes, in surgery, the patient's asleep, but they're not comatose. They're in a deep sleep and there's still muscle tone. You have to account for that. If somebody tore an Achilles tendon, giant mistake, terrible event to occur. If we tighten it too much when we sew it back together, or whatever means we use to fix it, all of a sudden the foot's going to be up like this. If we make it too loose, the foot's going to drop and flop around. You have to get it just right. So you have to account for that resting membrane. And that's what surgeons know over years of experience, the more or less 10 years of training after medical school, residency, fellowship, postdoctoral studies. It's a long time. So you get the idea of what's right and what's not. And it really takes a long time. So understand that it's a well-known process. So these end plate potentials are pretty much continuously occurring, and periodically they become action potentials, and that keeps the muscles in tone and sort of use it or lose it. That's very much contextual material going forward. That's there. And so that's what happens. And eventually we start with local changes. And then what happens if this change reaches threshold, it will spread all in every direction from where that is contacting the muscle, here, all over it. It's multi-directional. When we talk about nerves and we talk at the same phenomena, it's unidirectional. The impulse only goes, like you were seeing here, down the nerve. It doesn't go all directions. And we'll explore the reasons why that occurs. So you're going to see that. To summarize, the action potential arrives at the axon terminal. When it gets there, voltage-gated calcium channels open. Calcium enters from the outside. Remember, the muscle's got calcium in it to make it shorten. This is different calcium. This is extracellular calcium. The calcium releases the acetylcholine neurotransmitter. And basically, that's what it does. That's what happens with exo and endocytosis, how the little vesicles fuse. And remember, I told you that's the process where Botox works. Botox, for whatever reason, not clear, prevents that from happening. So as the acetylcholine diffuses across these acetylcholine receptors, sodium, they erroneously should call them sodium-potassium chemical gates. It's not just sodium. And it binds to those receptors, opens the gates, sodium flows in, potassium also flows out. And that's something I should have put in that as well. And what's in there, and it goes away right away. And so really what determines whether or not we reach threshold and whether or not that muscle fiber actually fires has to do with how much is released and how rapidly it's released, because it's continually degraded. The degrading phenomena involves a chemical called acetylcholine esters. So the acetylcholine are these little dots. Acetylcholine, I'll put the esterase down here because that's not how you spell it. The ester is the chemical structure. ACE is the enzyme, and it breaks apart acetyl and choline. And what it does, some of the acetylcholine will just go away very quickly by diffusion. The acetylcholine esterase will break it apart, break down metabolism, however, degradation, they have different words, and will separate it into acetic acid and choline, which are separate. And there's a lot of terms. I use metabolism or breakdown. I think this text uses degradation. And then lastly, the acetic acid we use all over the place. But the choline, we recycle. That process is appropriately called, but they miss it here in the animation, as reuptake. And really, it's very useful because it's sort of a microcosm of how neurotransmitters, including acetylcholine, are taken out of a synapse, whether they're in the nervous system or not. It's very specific. I really am emphasizing that reuptake process because it's when we use medications for psychiatric or psychological or nervous system disorders, where those medications work inevitably, if we understand them, and we don't understand all of them, is at the synapse. We either make things more likely to fire or less likely to fire. We impact enzymes that break them down. We impact reuptake. We impact all of those things. It's really, really important going forward. So you're going to hear, and that's something we cover when we talk about how the nervous system works on a microscopic basis. That's effectively when we conclude this rather thick chapter, we get to that chapter next. And so that's when they talk about the term degradation or degrading, it means breaks it down. So this is just showing, and they go over all these steps. It's very hard to read, but you can see, I mean, all of these things that are there. And then here they're showing the sodium, potassium. And then here they're showing the degradation. So rather than go crazy with that, quit calling. Drive me insane. You know I'm teaching. Stop doing that. Did I say that? Let me show you. Let me show you this. It'll be easier. I'll adjust the sound.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neurotransmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
That's the reuptake that they're talking about. And this is about done. So effectively, here it is. So it's and that's really microscopically, and I think we have some slides that demonstrate that. It just sits, it's almost like little feet, and even though it implies that they're contacting, but they're not. There is this space, a gel-filled space, we call it the synapse or synaptic cleft. It's not sealed like a chamber, so it allows the diffusion of stuff. So that's why it's a very rapid turnover process, and it's really well understood. And as you will see very shortly, for good and for bad, unfortunately. And now they're highlighting that for you. And here's the motor end plate. So you can see it's almost laying on there, but effectively not touching it uniquely. And so here's that signal. Here are the synaptic vesicles filled with acetylcholine. And then that voltage, what it does, it really doesn't affect them directly. It's indirect, because once the calcium flows in, it changes in such a way to allow these to be exocytosed. It involves something you don't need to know about, those V snares and T snares. Not going there. That's where Botox, not going there for you folks. But understand that's really the importance that's there. So here it is flooding this area. And there's just thousands and millions, actually millions, if you wanted to be technical about, given how small these little receptors are. They receive the acetylcholine, and since it's a chemical, they're called chemical gated or ligand, which is the role of the chemical. Either term works. Channels. And what they do is they're not just for sodium, they're for sodium and potassium. And as the illustration shows very nicely in the animation, it has more sodium in red going in than blue potassium going out. So it shows that to you pretty well. Sorry. So you can take a look at that part and what's going on. Now, as we come to an end of this, if it is sufficient, it will effectively generate, as you can see, the action potential going effectively all over. And I'll show you what governs that, and all that will do is confuse you more, unfortunately, but you need to see it. And then lastly, they talk about this remarkably rapid process of getting rid of it. Okay, so some of it can just go away because it's not sealed in that chamber. That's diffusion. And basically your body will break it down and use it for other stuff. It does break it down in there, and that's probably what it does more than anything else, into its component parts. Acetyl becomes acetic acid. We use that in all sorts of different chemical reactions. Acetyl groups are pretty common. And then the choline we use in a lot of other things as well. Inside the cell, making proteins and phospholipids. And I mean, there's just all sorts of stuff that choline gets used at and what it's attached to. But what the most important thing about the choline is it undergoes reuptake, or, this, they were describing this resynthesis. Basically, and this is common, most of the neurotransmitters, when we start talking about dopamine, when we start talking about serotonin, monstrously important neurotransmitters, we look at them being taken up. Okay, they effectively recycle them, and because of that, that's the area of concentration with a lot of medicines for minor depression, for eating disorders, for obsessive compulsive disorders, for mild bipolar disorders. Basically, they focus on inhibiting the reuptake. If you stop the reuptake, or if you limit the enzyme's ability to break it down, more is in there longer. And that causes not only for that circuit to keep working, over time you will see, and we get to this in that chapter on really how they work. It actually alters the receptor. It's fascinating stuff. So there's a ton to it that's there. And that's what they're showing you in that particular animation. This is the part where you start hearing about the good and the bad. Just like the rheumatoid arthritis we talked about with regard to true inflammatory arthritis, this is called myasthenia gravis. It's also almost assuredly an autoimmune disease. Rather than attacking the joints, these chemicals are attacking, something else. They're attacking, the receptors on that plate. And by attacking that, they don't work very efficiently. So what they came up with were things that inhibited acetylcholine. And so you were giving to them in doses that were workable, and the acetylcholine esterase rather. So they used to use the term ACE inhibitors. Probably shouldn't use that because acronyms are common and often spelled the same. So there's another system in your kidney and another class of pharmaceuticals called ACE inhibitors. So I stay away from that intentionally. So by inhibiting the acetylcholine esterase, you leave the acetylcholine in there longer, and because it's in there longer, those muscles don't tire. And it's always muscles you use the most. Your eyelids rapidly, every 5 seconds, you talk, we do, all the muscles we use a lot. And the way these people would start to, they can't keep my eyes open. And then eventually have trouble breathing, and then it's a real downhill spiral. So the good news is we have medicines for that. The bad news is if we took large amounts of this chemically active stuff and aerosolized it, we would create what is known as a chemical weapon. So when you hear about people being gassed with chemical weapons, that Saddam Hussein did to his own people in Iraq, that the ousted, trying to remember his name, the Syrian leader who was ousted, now living in Russia, did to his own people, that was first started in World War I. I mean, those are chemical weapons. We have them. They did it in Tokyo. Terrorists did that in a subway and released it. A lot of people died. That's VX gas, another nerve gas. They're nerve gases because they work on the nervous system, but they work here by inhibiting the acetylcholine. And they do it so rapidly. And what happens typically it affects respiratory muscles. We use a lot. And imagine somebody who inhales and can't possibly exhale. They fire it and they can't relax. It's a terrible way to die. Terrible chemicals. That's why they're weapons of mass destruction, and that's why they're outlawed. Not to say people don't have stockpiles of them, but we kind of go crazy when they use them. The trouble with science, and the good thing about science, we seek to understand processes. The vast majority of times we're trying to understand it for good, to help people, to evolve ourselves in the medical community, understanding a variety of things. The bad news is there are people who, will find ways to use it to hurt people. See it. So again, the whole idea, and we're getting to the end of today's presentation, the resting sarcolemma, the membrane, polarized, positive on the outside, negative on the inside. The whole ball game is rapidly switching it. Those end plate potentials, they have a short lifetime. They only rapidly switch it right by the end plate. But if there's enough of them, if we have enough neurotransmitter in a sufficiently short period of time, what will happen? It will cause the entire muscle fiber to begin to do that process. The name we give that is depolarization. Remember, it's polarized, positive outside, negative inside. If we flip it, we've changed the polarization from now to negative outside, positive inside. By flip-flopping it that way, we have changed it or depolarized. And when we reset it back to positive on the outside, negative on the inside, which happens, and these all happen in fractions of a second, we repolarize. And so when we depolarize, we're basically taking the interior of the cell along the membrane and shooting it up positively. When we repolarize it, it's going back down negatively, back to the resting. We call that an action potential. When you see the spike on an electrocardiogram, and you've all seen that, blip, blip, doctor show, whatever, real type show, doctor show on TV, and when it flatlines, it was a big trouble. That's the heart, and we're basically collecting all of the action potentials. That big wave going up is called the QRS complex. That is the big part of your heart, the big muscles of your heart, called the ventricles, depolarizing. It's about electrical activity. That's what this is leading into in AP2. So the end plate potentials are the opening of those chemically or ligand-gated ion channels. Sodium in, potassium out. It's always 3 in, 3 sodiums in, 2 potassium out. So the net effect is it's going to get more positive on the interior of the membrane. If it does so enough, it reaches something we call threshold. And threshold is like a tipping point. Once you hit that threshold, then that wave is going to spread. Why that occurs, you're going to hate this. Sorry. End plate potentials are generated. So here they are. What happens if they get far enough past the motor end plate, they strike these receptors. This is why you don't like me today. These are called voltage gated, first sodium and then potassium channels. What happens if it gets far enough? Not just a little sodium is going to go in. Massive amounts of sodium are going to go in and rapidly create that uptick we call depolarization. And right after that, almost instantaneously, fractions of a second, literally milliseconds, and you're going to see the graph. Right after it, that shock wave opens up potassium and it flows out. So the sodium goes in, large amount depolarizes, and the potassium goes out, repolarizes. Sodium in, potassium out. And when this all is said and done, and continuously during the process, those sodium potassium pumps are trying to rapidly reset the chemical balance. They're kicking back out 3 sodiums, they're pulling back in 2 potassiums. That 3-to-2 relationship applies here at the motor end plate and those sodium potassium pumps, except in opposite directions. Understand that electrical events happen rapidly, and even though you're resetting the cell electrically by sodium going in and potassium going out, you now have to reset it chemically so it can fire again. And this really becomes relevant when we start talking about the heart more than anything else. And we start talking about why nerves only go in one direction. There's a really important process. So depolarization, generating this, the end plate potential enough to that critical level we call threshold, the voltage gated sodium channels in, a large doesn't do it justice, through channels into the cell triggers an action potential that is, how RMU of it, unstoppable. I can't help myself. I do it every class. Drives me nuts. And will lead to muscle fiber contraction. The action potential spreads in all directions from one voltage gated channel to the next to the next. In the meantime, back at the ranch, right next to that voltage gate, all those voltage gated potassium sodium channels are potassium channels. They're going to start to correct it, basically the electrical aspect of it, by compensating for all that sodium coming in by kicking out a bunch of potassium. So that's how it flows in that direction, except in the muscle, it goes all over. It's a little different in nerves. That's when we're going to get to start looking at that. That's sort of like if this is a gate, when you close the gate and lock it, that's the lock. That's called an inactivation gate. And because there's a certain amount of time, what effectively happens is you create something we call an absolute refractory period. So at repolarization, when the sodium's closed, they also temporarily lock, just go with that. And even though potassium is going out, that muscle fiber cannot be stimulated. In nerves and muscles, it's a matter of milliseconds, 5, 7 in a muscle, less in a nerve. So, I mean, we can do this until we fatigue. It resets very quickly. And nerves even faster still. That refractory period is significant when you get to the heart, because before the heart can fire again, it has to rest for a total quarter of a second. Remember how rapidly it beats. A normal heart rate could be as low as once a second. So it's only beating, literally it's only beating for a quarter of those seconds. And then it has to rest for a quarter of a second. You get that when you talk about the heart. And so the ionic conditions, the chemicals, still have to be reset. That's the sodium potassium pumps. And they show it to you. So here, repolarization, restoring it. This is the graph. Sodium channels open. Bang. Peaks here. Depolarizes rapidly. This is rapid. 5, 7 milliseconds. Down here. So for this period of time, until this gets probably to about here, when it gets below sort of that threshold level, effectively we are in that what's called the absolute refractory period. You'll see it graphically in future chapters. It's not so relevant now. It's particularly important when you get AP2 to understand how the heart works, I think. And that's really what we're going to come to next. This is the end of today, the excitation, contraction, coupling. Have a great weekend. Good seeing you. All, whatever, less than 20 of you, but